So, Dr. Levin, I think had I known how important ion channels would be to the future of biology, I think I would have paid attention a little bit closer in cell biology class all those years ago. And what I also didn't appreciate or think that I would see in my lifetime is somebody coming up and sort of challenging the DNA-centric model of biology that we all really understand and appreciate, that's printed in every single textbook today. And that's basically that DNA is the arbiter of cell fate. It controls the whole protein inventory of the cell and runs as a sort of software. That's that analogy that we always hear. But you have a very unique perspective based on your work in planaria amongst other organisms. But I think in order to understand your thesis, we should start with planaria. What is planaria, and why is it such a fascinating organism?
Yeah, maybe if we could just take a quick step back, and I just want to say one thing about what you just said. So, actually, there've been a whole bunch of people in general arguing back and forth around the centrality of DNA and all of that, right? Most recently Dennis Noble and many people before him. So I'm certainly not the only one, but what we do need to think about, I think, is what do we really mean by this? In other words, when you say that a particular set of mechanisms or a particular level of description or whatever is that's the explanatory level, what do you really mean by that? And I just want to talk about that for a quick second. Absolutely. Because we're going to get into a bunch of other examples from our work and all of that. And I want to be clear that I'm not saying that genetics and genetic information isn't important. But I do have a specific criterion for what we mean by when we say these things are central. And what I mean by that is that we want formal models that facilitate discovery and facilitate invention. And this is important because I'm gonna describe to you a number of experiments, and everything that I talk about, one could look at that and say, oh, I think I can explain this in terms of genetics and biochemistry. Right. And this is true because much like with every act of human behavior and every physical event, if you wanted to drill down to the lowest level and look at the kind of the low level hardware that made it happen, yes, you will always find the chemistry and whatever quantum physics below that.
Right.
It's not you don't find magic, you don't find anything like that. My claim is not that when you look underneath these examples, I'm going to tell you you're going to find something other than the conventional laws of physics and chemistry. My point is that if you do start out with that as your primary level of description, there are a whole variety of discoveries, including some very useful things for regenerative medicine, for bioengineering, that they are going to keep you from finding. And right. So I just want to get that clear, is that looking backwards and providing reductive mechanistic explanations for things is always possible going backwards. But what we want to understand is what kind of worldview helps you discover these things, and what kind of worldview actually predicts some of these things as consequences.
Right. And I hope to get into this a little bit later. It's also a product, I think, about perspective and where you're coming from. As a molecular biologist, I dealt a lot with DNA transfections and that sort of thing. And you come from a computer science background. And so I think that has a lot to do with the way you approach the problem. We'll get into that later, but I think that's a little bit of what you're getting at as well.
Yeah. Okay. Yeah. Let's talk about planaria. So planaria are an amazing creature. They're a pretty popular system nowadays for regeneration and for behavioral pharmacology and for some other things. They are a free living flatworm. There's many species. It's a group of free living flatworms. They are anywhere from half a centimeter to a couple of centimeters in size. You can obviously see them with your naked eye. They are not like earthworms in that they are true bilaterians. So they have bilateral symmetry. They have a true brain. They have many of the same neurotransmitters that we do, and so they are similar to a kind of an ancestor of our lineage. So they have some interesting properties. The first is that if you cut them into pieces, every piece will give rise to a perfect little worm. So they have this amazing like holographic kind of property where they regenerate. They regenerate every part of their body. They can regenerate, and almost every part of their body can regenerate. There's a couple of small exceptions.
Right, and I think just the term holographic, I think, sometimes gets misconstrued with some woo stuff. What you mean by that, I think, is that every cell in the body has the information coded in it to make every other part of the body.
Correct. I mean, we'll talk about information and how you code information and all that, but functionally, what I mean is exactly this. I mean, holography is it's an area of engineering. It isn't a woo thing. It's what it means is that once what you have a material where you can cut it into pieces and you can recover the whole information.
Right.
You don't end up you don't end up recovering just the piece that you cut. Right. So that's all I mean by that.
Yep.
So yeah, so they have that regenerability. Also, they're smart. They can learn, so they can learn in a variety of training assays, which means that you can do behavior and brain regeneration in the same animal. That's very unique. That means you can train them on something, you can cut their head off, they regenerate a brand new brain, and then you can, for example, find out that they remember the original information. So that's pretty amazing, and that's not easily doable in most other systems, although there have been attempts. They are cancer resistant, so they have very, very low rates of cancer. They survive carcinogens and all of that. And the asexually reproducing ones, which basically the way they reproduce is they tear themselves in half, and each half regenerates. Right, the head makes a tail, and the tail makes a head. Those lines of worms are immortal. They don't age. So there's no such thing apparently as an old asexual planarian. So those are some amazing things. One implication of the fact that they reproduce asexually is that, unlike us, who go through a sperm and egg stage, so that if you get a mutation in your body, your children do not inherit that mutation. In planaria, that's not true. So any mutation doesn't kill the stem cell, gets amplified into the next generation because that cell has to rebuild, right, a bunch of body tissues. So isn't that wild? I mean, we just to start with that right off the bat, what in your biology education would have led you to predict that the animal with the noisiest genome that accumulates for 400 million years, has been accumulating these somatic mutations, is actually the one that's highly regenerative, cancer resistant, no aging. Right? Isn't that sort of opposite to everything that we've been told about the role of the genome and how all this works?
Yeah, absolutely. And something that unless you're deep in the world of sort of model organisms and studying, you don't often hear of planaria, period.
Right.
And so, yeah, fascinating creature. Absolutely.
Yeah, and they also another thing I forgot to mention. They are which finally, after years, this puzzled me for many years, and finally I think we understand what's going on. They also are not only resistant to transgenesis, so you can make transgenic flies, mice, whatever. There are no transgenic planaria, nor are there any planarian strains with abnormal morphology. So you can call the stock center, and you can get flies with curly wings and mice with weird tails and whatnot. Yeah, the only abnormal strain of planaria that you can get is our two-headed form. It's not genetic. And so there's actually something very interesting going on here, which we can talk about if you want, which is why is the genetic level not amenable to in planaria, not amenable to that kind of manipulation?
That's fascinating. The transgenic point there, I had never heard. That is something. Yeah. What are your thoughts on that? Because back to the DNA centric view of biology, that's something you'd never imagine.
Yeah. Well, so the way we make our two-headed worms is by changing the information in the bioelectric circuit that they have. So they have a stable bioelectric pattern within their tissues that says one head, one tail. And that pattern is a kind of memory, literally, because you can alter it with a brief physiological experience. You can change it. You can say, two heads. And from then on, all the pieces regenerate as two-headed in perpetuity. So the DNA has not been changed. And yet the pieces will continue to regenerate as two-headed. So that tells you right there that the answer to how many heads does a planarian have is not in any sense coded in the genome. What the genome does do is produce some hardware that by default self-organizes a bioelectrical pattern that says one head, in the same way that when you buy a calculator from the store, you turn it on by default, the first thing you see is zero, reliably 100% of the time. That's what it does. So yeah, so you make some hardware where the circuit is such that under default conditions it says one head. But like a reprogrammable kind of device, which I think these are, you can actually you can change that information. And so what I think is going on in this case is that and we've now done a bunch of computer simulations of this kind of process. It goes back to asking the question, how does evolution proceed with a very competent substrate. Most of the time when we think of evolution, when people model evolution, they go straight from here's your genotype. Your genotype determines the phenotype, and the mutation operator works on the genes. The selection works on the phenotype, and you put it in a cycle, and then we all know how that turns out. And people do genetic algorithms and things like that. What you're missing is the middle layer, which is morphogenesis. It's and the reason it's important is that morphogenesis is not a passive mapping from genotype to phenotype. It's a problem-solving intelligence, and I mean that quite literally. It's a system that is able to solve various problems. And what happens there is that just imagine for a second, and we'll get back to planaria. We showed a while back that if you have a tadpole, tadpoles have to become frogs and they rearrange their face. And what we showed is that if you scramble the craniofacial organs of a tadpole, you still get a normal frog. They will actually go through novel moves, novel movements, and eventually you get a normal frog. Right?
Problem solving.
So just imagine. Yeah, right. So navigating the anatomical morphospace. You have an anatomical space, and you're navigating from where you are now to where you need to be. And then we'll get back to how you know where you need to be, which is these bioelectric pattern memories. But getting there is not always easy because you may get taken off course. There might be a barrier. You might be broken in various ways. There's all kinds of things you might need to deal with. So just imagine, so you're a tadpole and you suddenly have a mutation, and most mutations do more than one thing. So you end up with, your mouth is off to the side of your head, but also something else positive is happening in your tail somewhere, right? Under the standard model where the genetics determines the morphology directly, that tadpole is dead. Your mouth is off to the side. You can't eat. That's it. And so now evolution can't explore the positive benefits of that, of the other positive benefits of that mutation. You have to wait until somehow whatever that feature is appears by itself without the thing that moves the mouth. That's very difficult in a pleiotropic model like this, where every gene affects multiple different things, and evolution should take a really long time. Instead, what happens is the mouth will fix itself. Okay, the mouth will actually move to where it needs to be. And then by the time this organism comes up for selection, that part's fine. And now you get to explore whatever else is going on with this mutation. So what's happening is that when the material over which evolution is operating is not passive, the way that we engineer with wood and metal, but it's not even active matter. It's actually, I call it an agential material, right? Because it has certain autonomous abilities to get certain things done despite novel circumstances. What happens is that it hides information from selection. When selection sees that tadpole, it can't tell that, hey, actually your genome wasn't very good. We need to weed you out because selection just sees the phenotype. And so by having these self-repairing properties and this ability to get to where you need to go, and we have lots of wild examples of systems that do that, you end up hiding genomic information from evolution. And when you do that, selection has a really hard time picking the best genomes. So it has a hard time seeing the structural genomes. Where does all the work get done? Well, all the work gets done on improving the competency. So instead of improving the structural genome, what you end up doing is improving the mechanisms that allow you to get the job done, even if your hardware is a little wonky. But that starts a positive feedback loop, because the minute you do that, you start generating even more competent tissues, which hide even more information from selection. And so you can see this in the simulations that we've done. Evolution running on this kind of on this kind of agential material acts completely differently. What happens is the pressure on the hardware flattens out, and then all the work gets done on the algorithm itself. So now just imagine you end up, and I don't know why certain lineages stop at different positions along the spectrum, but you've got a spectrum. On the left side of the spectrum, you've got something like C. elegans, which is incredibly hardwired, right? So every cell is numbered. Every C. elegans has exactly the same number of cells. They're all lineage determined. As far as I know, it doesn't regenerate much of anything. That's it. Then on the opposite end, you have planaria. In planaria, that ratchet went all the way forward because the material is so unreliable, because you're going to be accumulating mutations, right? That because of this crazy somatic inheritance that they practice, the genome, the genome's going to be noisy. So all the work went into making an algorithm that, for all intents and purposes, ignores a lot of the information in the genome. This is why transgenesis doesn't work. This is why we can't make mutant lines in planaria.
Wow.
for the same reason that the natural mutations that have been accumulating for hundreds of million years don't change. All the work has gone into that algorithm, and in between you have things like salamanders and then things like mammals that are sort of, they can make up for some things but not others.
Interesting. So it's as though it's as though this defense mechanism, for lack of a better term, evolved over time to protect this noisy genome, and that's why it's and that's why it's pushed all the way to the other side of the spectrum. That's fascinating.
I think that's true, and I think that in this particular case, that's why it's gone so far. But I also think that fundamentally, this issue of and this is really interesting, this issue of working with an unreliable substrate is true for all life from the very beginning. This is one of the ways in which computer. So I use a lot of computer analogies both in my talk and in my my discussions and in the research. But one way in which the current computational paradigm absolutely does not match biology is that in computer science, what you try to do is separate the layers. You try to encapsulate the layers so that when you're writing code at the high level, you don't need to worry about, well, geez, if the copper gets a little warm, all my registers are going to float off and the numbers will, start to, you don't worry about that. You assume that the hardware is going to do what it needs to do, and you're operating at a particular level. Biology, I think, commits from the start to an unreliable substrate. You never know what you actually have. You're guaranteed to mutate over time. You don't know what never mind the environment, but you don't even know. And again, there's many amazing examples of plasticity in biology where wrong copy number of your genome, it's fine. The wrong number of cells, also fine. Wrong cell size, no problem. They figured it out because even normal embryos under normal circumstances, we're not assuming that everything, except maybe C. elegans and things like it, we're not assuming that everything was going to go correctly. They are problem solving in real time every single time, and that's what accounts for the incredible plasticity. And in some cases, like planaria, just it like takes off exponentially.
That's fascinating. Do you know, is there a correlation between genetic or genome noisiness and this ability to regenerate? Is that something set in stone?
It's a good question. I don't know if a good study across phylogeny has been done. I'm not sure. I mean, it's probably information about, for example, axolotl genomes and things like that. Yeah, that could be done. I'm not sure. I'm not sure that's been done.
And then sitting here as someone who did a lot of tissue culture work, you're talking about transgenic. Making a transgenic planaria is virtually impossible. Can you ectopically express genes in not even ectopically for a moment?
It doesn't seem. I mean, it's entirely possible that somebody will crack this. People have been working really hard for 40 years on this now, and nobody's really done it. Maybe somebody will crack. I mean, presumably there's a way to overcome it, right? There's got to be, in terms of synbio and whatnot, there's got to be some way to overcome it. But the natural situation is very interesting. It's just not taking that information as literally as other systems do.